Hello students, welcome to my YouTube channel. Hope you're all doing good. In today's session, we shall understand a topic in biochemistry, that is ornithine cycle under the catabolism of amino acids. I'm Dr. M. Bhushanam, Associate Professor in Zoology, Maharani Science College for Women, Bengaluru. The learning objective of today's study is to understand the process of ornithine cycle of amino acid catabolism and also the diseases associated with the deficiency in the performance of this chemical enzymes. So content includes, under the catabolism of amino acids, the concept of ornithine cycle. Well, students, as we have understood in earlier sessions, the polypeptide chain or a protein chain undergoes degradation or hydrolysis to form unit molecules referred as the amino acids. The dietary proteins that we consume will break down ultimately to the units of amino acids, and these amino acids further degrades through three major reactions. Number one is the transamination. In this particular reaction, there is transfer of the amino group from one amino acid to another amino acid takes place through the catalysis of the enzyme referred as transaminases. So, the process of transamination involves two amino acids, one that donates the amino group, the other one that receives the amino group. The one which donates the amino group is called alpha amino acid, and the amino acid that receives the amino group is called alpha keto amino acid. Students, this is the first step under the degradation of amino acid. The second major step or reaction is again related to the amino group itself, and this step we call it is deamination process. It's a stage wherein the amino group gets eliminated or removed completely from an amino acid. So this process we call it as deamination. This reaction again involves a group of enzymes called as deaminases. The third important chemical reaction refers to decarboxylation, where the carboxyl group of the amino acid is removed, and it also includes a group of enzymes called decarboxylase enzymes. Now, overall, the amino group to have the process of degradation, they tend to show the removal, either the transfer-amination process or deamination process. Both the process shows the reaction with that of the amino group in general. Now, what's happening to the amino group here? The amino group is eliminated freely in the cytosol of mitochondria or a cytosol of the cell, and this amino group, when it remains freely, then it is very dangerous to the cell as well as for the organism, because it causes tremors, it causes inactiveness of the nervous system, and it also causes death of the cell or sometimes even to the coma state of the organism. So in order to overcome this toxic substance presence in a cell called the ammonia, we need to immediately throw it out. So the accumulation of the toxic substance of ammonia in a cell is very, very dangerous for the cell because it kills the cell, it kills the organism. So what's happening here overall? The amino group that is getting transferred into ammonia, if it gets retained within the cell, that causes the danger. So this immediately is converted into less dangerous or less toxic chemical called as the urea. So ammonia is more toxic, urea is less toxic. Ammonia also contains amino group, urea also contains the amino group. So what's happening to the amino group, which is formed in the form of ammonia, which is more toxic and dangerous for the cell, will be immediately converted into less toxic ammonia form called as the urea. So this urea formation that happens within the cell or in the organism is referred as urea synthesis. Students, remember, the synthesis of urea always takes place in a cyclic process, and hence it is called urea cycle. Now it is referred as ornithine cycle. So what exactly ornithine cycle refers to? It refers to the cyclic chemical reactions wherein the urea gets formed from the ammonia. So urea is the ultimate end product of the protein or amino acid catabolism. Remember, in this stage, the ammonia that contains nitrogen group, which is very toxic, will be converted into urea, which is less toxic. So this process of removal of toxic substance from the body of a cell or of an organism, we call it as detoxification. D means removal. So removal of toxicants or toxic products is called detoxification. So urea cycle or urea formation also refers to a process of detoxification of ammonia. Now, this nitrogen-rich urea, once if it is formed, it will be formed in the sides of the liver cells or hepatocytes. Later will be thrown into the bloodstream. From the bloodstream it reaches the kidneys. In the kidneys, nitrogen-rich urea will be excreted out in the form of urine. So during the urine formation, the urea will be expelled out of the body of an organism. Students, this urea formation takes place in the liver and transported to the kidneys for excretion process in the form of urine. So this is the importance of the ornithine cycle or urea formation that we are going to understand in today's session. The process of formation of urea, when we look at it, is a cyclic process that includes a number of chemical reactions, which is together or collectively known as ornithine cycle or urea cycle. Ornithine cycle is the name that we give recently for the same process of urea formation. Earlier it used to be called as urea cycle. And this cycle was explained by two scientists, namely Hans Krebs and Kurt Hensleit in 1932. And therefore, this urea formation or urea cycle or ornithine cycle is also referred to as Krebs-Hensleit cycle. So down is the picture. Towards left side is Krebs, the right side is Hensleit. Well, students, chemically urea consists of two amino groups, which is formed from the reactants, namely ammonia and carbon dioxide. So ammonia with carbon dioxide and ATP molecules, it forms the unit referred as the urea and water. And this happens in the organ of a liver in the body of an organism. Students, when we look at the urea formation, overall catabolism of amino acids, where transamination, deamination and urea formation takes place within the liver tissue, because the enzymes required for the reactions of chemical that are involved in the urea formation are seen localized in or located in the liver, and thereby the process of urea formation also takes place in the liver tissue. So after the urea formation, it will be thrown into the blood system of the individual. From the blood, it reaches the kidneys, and in kidneys, the urea gets filtered and it will be thrown out in the form of urine. So this is the importance of amino acid catabolism, especially referring to the urea formation. Now, the synthesis of urea takes place in 5 major steps, which are cyclic processes, and each of the steps makes use of a very distinctive enzyme for this step, which are present in mitochondria as well as the cytosol of the liver cells called hepatocytes. To name them, the first step is synthesis of carbamoyl phosphate. The second step is formation of citrulline or L-citrulline. The third step is synthesis of argininosuccinate. The fourth step is cleavage of argininosuccinate. And fifth step is formation of urea. So the actual formation of urea takes place in the last step, but prior to that there are 4 major steps that involves in the synthesis of the urea. The first step to start with is the synthesis of carbamoyl phosphate. So what's happening in this particular step? Here, the carbon dioxide and ammonia ions will combine to form carbamoyl phosphate in presence of ATP. So ammonia, carbon dioxide and ATP molecules, all these 3 together combines to form a complex referred as carbamoyl phosphate, and this makes use of an enzyme referred as carbamoyl phosphate synthetase or synthase enzyme. So the reaction of synthesis of carbamoyl phosphate has the reactants, namely ammonia, carbon dioxide, ATP molecules to form the first product referred as carbamoyl phosphate in presence of enzyme carbamoyl phosphate synthetase. Why are we giving the name synthetase for this enzyme? Because it is the enzyme that is forming the complex of carbamoyl phosphate from that of the ammonia and carbon dioxide. Now down is the reaction where you can find ammonia combining with carbon dioxide and ATP molecules to form carbamoyl phosphate, but of course it takes place in presence of enzyme carbamoyl phosphate synthase or synthetase. So what's this particular reaction, this particular step is the deciding factor for the rate of ornithine cycle or urea formation. If this particular process slows down, students remember, students remember, here the process of urea formation is decided by the carbamoyl phosphate synthetase and its enzyme. If this step is slowed down, overall process of urea formation also slows down. So this is the a step that decides the rate of urea formation in a cell. So here is a chemical reaction that shows the process of carbamoyl phosphate synthetase, where carbon dioxide ammonia in presence of ATP molecules forms carbamoyl phosphate. Step 2 is formation of citrulline or synthesis of citrulline. Students, in this step, the product of first step is carbamoyl phosphate, isn't it? So that carbamoyl phosphate will now combine with another compound present in the liver cell called ornithine. So this ornithine is already present in the liver cells. Carbamoyl phosphate is newly formed. Both are combined to form a product which we call it as citrulline or L-citrulline. So L-citrulline is getting formed from the carbamoyl phosphate and the units of ornithine. So this reaction makes use of the enzyme ornithine transcarbamylase. So, or simply it is also referred as carbamylase enzyme or carbamoyl transferase enzyme. So what is the importance of this? It is the enzyme that joins the ornithine with that of the carbamoyl phosphate group to form the chemical complex referred as citrulline. So here is a chemical reaction that shows how the citrulline compounds are formed. So remember, in step 1 you will find the formation of carbamoyl phosphate. In step 2 we find formation of citrulline. So citrulline is the combination of ornithine and carbamoyl phosphate together in presence of ornithine carbamoyl transferase enzyme. Now, the third major step that involves is again synthesis of the third important chemical reactant referred as argininosuccinate. So step 1 carbamoyl phosphate is formed, step 2 citrulline is formed, step 3 argininosuccinate is getting formed. This reaction also makes use of the product of the second step called L-citrulline that will combine with another amino acid present in the liver cell called aspartate. So aspartate and citrulline both gets combined to form a complex referred as argininosuccinate. So this reaction makes use of the energized molecules called ATPs, and it is also catalyzed by the enzyme argininosuccinate synthetase. So synthetase enzyme is used related to the production of argininosuccinate. So wherever we find synthetase enzyme, students remember that forms the product there. So based on the synthetase, we also talk about the kind of product that gets formed here. So down is the reaction where citrulline, ATP and aspartate, all these three molecules combines to form argininosuccinate. So of course in presence of argininosuccinate synthetase enzyme. So here is a chemical reaction that shows how aspartate and citrulline combines in presence of argininosuccinate synthetase enzyme by using ATPs that forms the argininosuccinate. So what's happening in each of this chemical reaction? Synthesis of a newer chemical compound is getting formed, during which the ammonia units are transferred from one chemical complex to another chemical complex. So ultimately here in the third stage, we find argininosuccinate having the amino group on it. Now, step 4 is not the synthesis, instead it is breakdown. Here what's happening? Cleavage of argininosuccinate takes place. Otherwise, we can say it is the hydrolysis of argininosuccinate chemical compound to form the two unit parts called as arginine or argin and fumarate compounds. So arginine compounds and fumarate compounds are getting formed from argininosuccinate in presence of argininosuccinase enzyme. So there is breakdown of argininosuccinate to form units of fumarate and arginine or argin. So down is the chemical reaction here. What happens to this fumarate then? This fumarate is an amino acid that enters directly into TCA cycle and gluconeogenesis process. And what is left behind? It is only the arginine. Students, remember, this argin or arginine is the amino acid compound which is responsible for the actual formation of the urea in the last step. So here is a reaction where argininosuccinate breaks to form fumarate and arginine in presence of argininosuccinase enzyme. Now, the last step under the synthesis of urea is formation of urea or synthesis of urea itself. So in all the 5 steps, 4th step alone involves the breakdown. In the rest of all the 4 steps, other than the 4th step, we find in rest of all the 5 steps, the synthesis of the chemical. So in the last step, what is getting formed? The ultimate product that we require called the urea. So what's happening here? Here, there is the synthesis of urea takes place wherein arginine combines with the water. So we call it also as hydration of arginine. So when water molecules combines with arginine, it forms the chemical complex referred as urea as well as ornithine. So ornithine and urea gets formed from arginine plus water. So that's what we see down in the reaction. But this reaction is catalyzed by the enzyme referred as arginase or urease. So what's the function of urease enzyme or arginase enzyme? This is the enzyme which is responsible for the synthesis of urea, and at the same time this arginase will make use of water or water molecules. The catalysis of or the rate of reaction of arginase enzyme, when we look at it, is dependent on 2 major compounds. One is carbon dioxide, then the other one is manganese ions. If there is concentration of arginase is high in the liver, the urea formation definitely takes place there. When is arginase enzyme will be more? When there is ultimately the concentration of arginine is more. At the same time, they are also dependent on the carbon dioxide and manganese units. And this urease or arginase enzyme is present only in the liver tissue, so urea formation ultimately takes place in the liver. So reaction wise, when we look at it's here, we find arginine with water in presence of manganese and arginase enzyme forms ornithine and urea. So this ornithine again will combine with the carbamoyl phosphate units to form argininosuccinate unit later. So it's a cyclic process where ornithine is reutilized. Now what happens to this urea? Ornithine, no doubt, is reutilized by the cyclic process, whereas urea will be expelled out into the blood, and through the blood it reaches the kidneys. In the kidneys, along with the urine formation, urea is also removed and it is sent out of the body system. Now, the overall reactions and energetics of the ammonia getting converted into urea, when we look at it, is like this. Ammonia, carbon dioxide and ATP molecules combines to form the chemical of urea fumarate units. Now here is a chemical reaction. How exactly the process of ornithine or urea gets formed? Students, remember, this is the chemical reaction that you should practice for the exam to represent. In step 1, in mitochondria, what's happening? Carbon dioxide, ammonia and ATP molecules are combined to form carbamoyl phosphate that you see towards the top of the left side of the cycle. Then it forms citrulline. Citrulline will combine with the aspartate to form the unit molecules in presence of ATP, argininosuccinate. Now this argininosuccinate will dissociate to form arginine and fumarate. Now arginine will be combining with the arginine will now combine with the water molecules to form the urea and ornithine. Ornithine goes back into the cycle, whereas urea is get expelled through the blood system in the kidneys in the form of urine. So this cyclic process is what you are supposed to remember and write in the examination also. So here is the chemical nature of the urea formation or urea cycle. Now, so after the understanding the actual concept of urea formation through ornithine cycle, we also shall have a knowledge of significance of urea cycle in the body of an organism. You know that every day in an adult healthy body system, 10 to 20 grams of ammonia is expelled in the form of urea. If there is problem with the or dysfunction with the enzymes involved, then the urea cycle process gets reduced by its rate or time. So slow process of urea formation takes place, and this leads to accumulation of more of ammonia in the blood cells of the individuals or in the body cells of the individuals, and this condition we call it as hyperammonemia. So it is a very dangerous condition where it can take to ultimately the death of the organism. If you look at the symptoms related to this disease of hyperammonemia, we have the condition referred as reduced growth or growth retardation, hypothermia, where the temperature of the body comes down. Then they will have muscular coordination, which is very poor. Then they suffer from dysdiadochokinesia. So it's a disorder wherein there is inability to perform fast muscular movement or activities such as fast finger tapping or foot tap, or sometimes even closing of the fist. All these functions will be slowed down in this affected individuals. Then they also suffer from hypotonia. It's a condition where the muscle tones of the body will be very poor. Then they also suffer from combativeness. It's a condition where it is related to the emotions of the mind, where the individuals will be eager to fight. Then they also suffer from condition referred as papilledema. It's a condition where there will be swelling around the optic nerve and optic disc related to the eyes. And these individuals also suffer from the blood poisoning due to the bacterial infections and bacterial toxins referred as septicemia. Then the affected individuals will also suffer from a disorder referred as ornithine transcarbamylase deficiency. It is due to enzyme deficiency in their production. It leads to a disorder referred as anorexia, where there is loss of appetite for having food. Then they also suffer from hypotonia. Then they suffer from vomiting, seizures, and then they suffer from somnolence. It's a condition where excess of sleepiness will be seen in this affected individuals. Then they also suffer from a condition referred as encephalopathy condition, where the function of the brain gets affected. Then they also suffer from immunodisorders and liver failure, which we call it as a cirrhosis, where there is the degeneration of the liver cells and the inflammation of liver cells are commonly seen. Then they also suffer from disorders referred as hemochromatosis. It's a condition where iron salts are deposited in the tissue and makes the tissue damage, which is commonly seen in the patients of diabetes. So these are all major symptoms related to the disorder. Well, students, with this we are completing the concept of ornithine cycle or urea cycle. And what is the outcome of today's session? We all have learned about the process of ornithine cycle and the various diseases associated with the defective enzymes of ornithine cycle. Well, students, after we understanding the concept of urea cycle, let us answer these MCQs to test our knowledge of understanding. The first question is, urea is formed from which toxic material? So what is the starting material which is toxic for the organism from which the urea gets formed? Definitely it is ammonia. Answer is B. Where do we find the urea cycle process that occurs? It is in the liver cells, so answer is B. Which of the following is the first step reaction for the urea cycle to facilitate its rate? When we look at it is carbamoyl phosphate synthase enzyme, and synthesis of carbamoyl phosphate is important. So that's the first stage that will combine ammonia, carbon dioxide and phosphate group. It is not given here in first three, so answer should be D. None of the above. Fourth question. Which of the following is the rate limiting step for the urea cycle? So definitely it is B, synthesis of carbamoyl phosphate, because that's the first step that will initiate and trigger the process of urea formation. Then, how many ATPs are required for the formation of carbamoyl phosphate alone? So it takes 2. So answer is A. So with this we are completing the concept of urea cycle and ornithine cycle. Students, the references that we made use here includes the web references of Wikipedia and Britannica.com, and book references includes the Animal Physiology Biochemistry by Dr. K.V. Sastry and Biochemistry published by COSIP of Bangalore University. Thank you all, students.